So the last thing I want to discuss in this lecture are the characteristics of the brain that makes us human. So those things that allow us to speak, to make plans, and to create music, for example. One of the differences between our brains and the brains of other primates is what is known as hypermorphosis, and that refers to the fact that for the first 2 years of life, our brains continue to grow at the same rate at which they do during embryonic development. So if you take a look at this graph, which shows the body weight and the brain weight for humans and chimpanzees, you can see that during development, our brain to body weight ratio and the growth is not that different. However, after we're born, our brains continue to grow for the first 2 years of life so that we end up with a brain to body weight ratio that is much higher compared to other primates. So during those first 2 years of life, we're adding a lot of neurons. Approximately 250,000 neurons are added per minute. And we're also increasing the number of synapses or connections in our brain. So this is one of the reasons why we have such a large brain to body weight ratio. Another difference between our brains and the brains of other organisms is the degree of cortical folding or the degree of gyrencephaly. And gyra and sulci refer to the hills and valleys of the brain. So the gyra are the hills, the sulci are the valleys. And if an organism has a high degree of cortical folding, it's said to have a gyrencephalic brain. Organisms like mice, which do not have cortical folding, are said to be lissencephalic. And the degree of cortical folding is generally, but not always, associated with the level of intelligence. And this is because there are organisms like elephants or dolphins, for example, which have a high degree of cortical folding or a high degree of gyrencephaly. But we could argue that dolphins and elephants are not smarter than humans. I mean, they're not writing literature, singing, creating music, or buildings, for example, that we know of, right? But this degree of gyrencephaly is not really thought to be associated with the number of cortical neurons in the brain. So the number of cortical neurons is mostly what's linked to intelligence, because the cortex is the region of the brain that's associated with reasoning and intelligence. And although elephants have a very large brain and they have a greater degree of gyrencephaly, they actually have less cortical neurons. The majority of their neurons are in the cerebellum. And if we look at the relationship between the folding index, so the degree of gyrencephaly, and the number of cortical neurons, we see that there's a poor correlation here, because humans have more cortical neurons, but they have a lower degree of folding compared to elephants, which have less cortical neurons. What is thought to be associated with the degree of cortical folding or gyrencephaly is the number of outer radioglia. And as you can see, these are shown here in yellow. And a mouse, which is lissencephalic, so it does not have cortical folding, has very few outer radioglia compared to humans, which have many outer radioglia. So these are thought to be providing the force for creating these gyri and sulci. In addition to that, the outer radioglia also express genes like ARH, GABP11B that are thought to be associated with the formation of the sulci. And no, you don't have to remember this name, but the important thing to note is that these cells, this outer radioglia, express these genes. And if we were to force the expression of these genes in brains that are lissencephalic, like the mouse brain that's shown here, we can see that these brains will form the gyri and sulci. So the degree of gyrencephaly will increase as a result. Another difference between us and other organisms like the chimpanzee is our genome. And even though I'm sure you've heard the phrase, we share 99% of our genome with chimpanzees, the reality is that we have other non-coding regions in our DNA, like non-coding RNAs, enhancers, silencers, promoters, that play an important role in the genes that are expressed in our brains. So for example, there are non-coding RNAs that we have that other organisms do not have, and there are regions that have been deleted in our genome. And one of these regions that has been deleted is the enhancer for the gene GAD-D45G, and this is a gene that inhibits brain growth. And because we have lost the enhancer for this gene, our brains are able to grow more than the brains of other organisms. And finally, another difference between us and other primates is the degree of gene expression that's happening in our brains. So if we compare the chimpanzee, which is shown here in the darker blue, and humans, and we look at the level of gene expression in different tissues, we see that for the blood and for the liver, that's pretty similar. But when we look at the brain, we see that our level of gene expression is almost 6 times greater than that of the chimpanzee. So these differences in gene expression are thought to be important for the characteristics that make us human. The very last thing I want to discuss is the changes that happen as we get older, particularly during puberty. And after puberty, the brain growth ceases and there is an increase in the amount of synaptic pruning. So neuronal connections are lost, and that's thought to be associated with our decreased ability to learn new languages as we get older. Another change, which you can see in these figures, is an increase in the degree of myelination. So this is showing the myelination in the darker blue, and as you can see, as we get older, the degree of myelination increases. And this happens particularly rapidly during puberty, but the myelination is not happening at the same rate in all the regions of the brain. So regions of the brain that are associated with fear versus those that are associated with reasoning may not become myelinated at the same time. So that might be, or maybe behind the reasons why teenagers may have strong reactions to stressful situations compared to adults who are able to handle stress a little bit better.